Okay, so we will discuss about the glucagon physiology. Glucagon is produced by as we have seen earlier, glucagon is produced by the alpha cells located peripherally with the isolate of the Langerhans. It is located as this is the isolate of Langerhans in the pancreas. This alpha cells located peripherally, okay, alpha cells, and this whole is the isolate of Langerhans. And this is the pancreas. Okay, so this alpha cells located in the peripherally in the isolate of Langerhans and in the endocrine tissue of the pancreas. Okay, in the endocrine tissue of the pancreas, glucagon release patterns tend to be the mirror image of those of the insulin. Okay, glucagon release is the mirror image of the insulin. Low blood glucose initiates glucagon release. Directly, low blood glucose initiates the glucagon release directly and also drive uh, nervous and hormonal release of the catecholamine, which activate beta adrenoceptors on the alpha cells to augment glucagon release. Okay, so glucagon release pattern is same as the mirror image of the insulin. Low blood glucose, when there is the low blood glucose, initiates the glucagon release and directly and also drive nervous and hormonal release of catecholamines, which activate beta adrenoceptors on the alpha cells to augment the glucagon release. Okay, so glucagon acts on the guanosine triphosphate binding protein, G protein. Okay, glucagon act on the uh, guanosine triphosphate binding protein, G protein. This we have to remember, that glucagon acts directly on the glucagon triphosphate binding protein, G protein coupled receptor. Okay, guanosine triphosphate binding protein coupled receptor that stimulate the production of the intracellular cyclic AMP. Okay, so this G coupled receptor, G protein coupled receptor, okay, will stimulate the production of the intracellular cyclic AMP in the liver cells. This results in the inhibition of the glycogen synthesis and the activation of the glycogen breakdown system. Okay, similar effect are obtained in muscle cell to increase the circulating level of the glucose. Okay, so how glucagon work? It act on the G protein coupled receptor, which stimulate the cyclic AMP. Okay, so we can write here that glucagon will act on the G protein coupled receptor, then will stimulate the production of the CAMP. Okay, what it will do, that in the liver cell and in the uh, muscle cells, it results in the inhibition of the glycogen synthesis and activation of the glycogen breakdown. The glycogen will be breakdown and the synthesis will also decrease. Okay, so this will result in the increase the glucose. Okay, by this mechanism glucagon will act. Okay, there are interaction between the glucagon and insulin within the isolate. Insulin inhibits the release of glucagon. Okay, insulin inhibits the release of the glucagon, but glucagon stimulates the release of the insulin. Okay, understood. Now that the glucagon and insulin interact in between the isolate. How? So if the insulin will increase, insulin will cause the decrease of the glucose. So as insulin will increase, it inhibit the release of the glucagon, will inhibit it. Okay, but as the glucagon as the insulin as the glucagon will increase, because it causes the production of the insulin, will increase the synthesis of the increase the production of the glucose, will increase the synthesis of the insulin. That's why as we give the insulin, the glucagon will is also decreased, so the production of the glucose will decrease further. And so in the diabetes mellitus patient, the hypoglycemia is very usually seen. Okay, then comparison between the insulin and the glucagon. Comparison between the insulin and the glucagon. So what happens that cell type, beta cells and alpha cells, factors that increase the secretion, factors that decrease the secretion, and mechanism of action and major action, and lastly the overall effect on the blood level. So uh, factors that increase the secretion from the beta cells, there is the insulin secretion is there, it causes increased blood glucose. Factors that increase the secretion, so when the blood glucose will be high, then amino acids, fatty acids, glucagon, secretin and acetylcholine, when they are high in the blood, the insulin will be secreted. Okay, increased blood glucose, amino acids and fatty acids when they are high, and the glucagon, secretin and the acetylcholine when they are in the blood, the insulin secretion will be increased. Then glucagon, so from alpha cells, it will secreted when there is a decrease in the blood glucose, increase amino acid, cholecystokinin and catecholamines and the acetylcholine. Okay, so when decrease in the blood glucose and increase in the amino acid, when there is the catecholamine, cholecystokinin and the acetylcholine, the glucagon secretion will be there. Then factors that decrease the secretion. So when there is the blood glucose will be low, somatostatin and the catecholamines, okay, decrease in the blood glucose, somatostatin and the catecholamines, and at this time the insulin secretion will be decreased. And when there is an increase in the blood glucose, insulin, somatostatin and the fatty acids and the keto acids, the glucagon secretion will be restricted. The mechanism of action, acts on the tyrosine kinase receptor to activate intracellular pathway that results in the translocation of GLUT4 transporter to plasma membrane. Okay, acts on the tyrosine kinase receptor. Okay, insulin act on the tyrosine kinase receptor to activate intracellular pathway. Tyrosine kinase receptor to activate the intracellular pathway that results in the translocation of the GLUT4 transporter to plasma membrane. Okay, GLUT4 transporter to the plasma membrane, acts on the G protein coupled receptor. It will act on the G protein coupled receptor to stimulate the production of the CAMP. Okay, will act on the G protein coupled receptor, which will stimulate the production of the CAMP. Then major actions, so increase the glucose uptake into the cells, then increase the glycogenesis, then decrease the glycogenolysis, decrease gluconeogenesis, increase protein synthesis, decrease protein degradation, then increase fat deposition, decrease lipolysis, decrease keto acid production, increase potassium uptake into the cell. Okay, this will done by the insulin, and by the glucagon decrease the gluconeogenesis, increase glycogenolysis, increase the gluconeogenesis, decrease fatty acid synthesis, increase lipolysis, and keto acid increase keto acid production. And overall effect on the blood level, decrease glucose, decrease amino acid, decrease fatty acid, decrease keto acid, and decrease potassium. Okay, and glucagon will increase the glucose, increase fatty acid, and increase keto acids.